All right. All right, everyone. So let's get going on this. Welcome to a virtual symposium, Metabolism and Cancer from Bench to Table. This is a joint venture by the University of Colorado Cancer Center and NORC. And NORC is the Colorado Nutrition and Obesity Research Center. And so we're collaborating both in terms of this symposium and also in terms of the grants program, which I'll describe really quickly in a minute. But our goal here is to really stimulate interactions and we recognize that we have a strength in metabolism on our campuses, meaning the entire cancer center, including CSU and CU Boulder, and that we needed to capitalize on this strength. And part of what we want to do here is stimulate interdisciplinary research, because there might be people working in basic areas who aren't really thinking about some of the potential applications of their work. And at the same time, there might be people working at much more applied areas, whether it's clinical or exercise or nutrition, who might be able to collaborate with basic scientists to try to, understand some of the mechanisms underlying what they're seeing in the real world. So with these talks, part of the goal is to stimulate these interactions. And so I've asked everyone to put up a sort of call for help slide. And so hopefully everyone's done that. And the goal of that slide is to sort of say, here's what we're doing, but here's where we kind of need help. These are areas that we don't have expertise in, or maybe you could use some resource, or, maybe you don't know where to find metabolomics, and we can certainly get people that can direct you there. So those are some of the goals. There's also an associated pilot grant project. Obviously, this is not limited to speakers. I've had a few questions about that. This is for anyone, and the RFA went out, so the details are there. One of the applicants needs to be a cancer center member, but the goal here is to stimulate interaction. So these proposals should be interdisciplinary. They shouldn't be completely established projects that are already funded. They can be projects that are already rolling along, but that have not gotten, large scale funding. Those are all fine. They're going to be 1-year awards, and we expect to give out 3 to 4 of them at on the order of 40 to 50K each. And again, this is with a buy-in from all 4 of our cancer center programs, from NORC, and from the AD for basic research, Haida Ford, and the AD for population sciences, Kathy Bradley. So this is, really we're leveraging different groups on campus to try to really goose our cancer and metabolism research here. So without further ado, and everyone should have the details in the RFA, I'm pleased to introduce Drs. Paul McLean and Ryan Marker. They're going to kind of tag team their presentation, which is going to be an overview of NORC cores and the BeFit Be Well program. So thanks.
Great, thanks, James. And I'm just going to share my screen here real quick. And hopefully you can see that. Okay. All right. So I'm going to talk a little bit about the NORC, as James said. Tell you a little bit about this is the Colorado Nutrition Obesity Research Center. I'm going to tell you who we are and what we do. A little bit about our programs and cores that will hopefully try to connect with you to some of the researchers that we have in our center with this interdisciplinary program. I'm going to talk a little bit about the programmatic development that we have going on that's related to that might be relevant to cancer researchers as well. So the NORC is an NIDDK sponsored P30 grant. Its mission is to promote nutrition and obesity related research. And this includes interdisciplinary collaborative work with a number of different disease pathologies related to obesity, like cancer. Our central theme is the prevention and treatment of obesity and its metabolic complications across the lifespan. We are one of 11 centers across the United States that are funded, and our NORC has members at 5 institutions in the Rocky Mountain region, but we collaborate with institutions all across the country. The research base, we have 120 members that are regular faculty. This has been growing over the past 25 years. And we have over 60 affiliate members who are trainees, faculties, scientists, and educators. So we have a fairly large group of people and a very diverse funding portfolio. About half is NIDDK, but because obesity and nutrition and metabolism relate to other diseases, we have funding from other institutes, including about 10% of our grants that are tied to the National Cancer Institute. This is our operating structure in the NORC. Our research base that I mentioned is supported by an administrative core, a pilot feasibility program, an enrichment program, and 3 biomedical research cores. Our faculty have studies in a diverse number of areas, including early life influences, metabolic programming, women's health, exercise and energy expenditure, metabolism and metabolic dysfunction, and interventions for better health. And people who do in our research base who are studying various types of cancer fall into these 2 categories of interventions to improve health and studies of metabolism and nutrition as they relate to cancer. Our pilot feasibility program is an annual program that particularly targets fellows and junior faculty that have not received our level funding yet. We welcome pilot award applications from Cancer Center members. We have a strong track record of success. You can see the number of outcomes that we're really building the future leaders of our NORC with this pilot and feasibility program. We've already gone through the process, but keep an eye out for next March or April when the announcement comes out, because we will welcome obesity and nutrition, metabolism, cancer types of projects with that program. Our enrichment program is led by Ed Melanson, and this we support a few formal seminar programs, but we really try to target existing enrichment programs with with our enrichment program. So partnering with people like the Cancer Center to bring nutrition and obesity researchers to their existing enrichment programs is something that we're very interested in. But something that might be helpful to a number of you who are listening is that we support a number of journal clubs and interest groups that many of which could have relevance to various types of cancer. Everybody's welcome to these. And for a list of these smaller groups who are interested in metabolism, nutrition, and obesity, you can contact Ed Melanson, who directs our enrichment program. Our research cores are meant to span the translational ladder. The molecular, cellular, analytical core is at the basic level. Energy balance assessment core fosters research in preclinical and clinical research. And then we have a clinical intervention and translational core, which is at the top of the translational ladder. Our molecular core is run by Brian Bergman, Kartik Shankar, and Matt Jackman. And here we have expertise and equipment for isotope enrichment studies, quantitative lipidomics, with the collaboration of the Cancer Center. Kartik runs a cradle-to-grave RNA-seq pipeline. And then we support a number of pieces of equipment that many of the Cancer Center, many of the end users are Cancer Center members. This equipment is for studies of metabolism with the Seahorse, the Ouroboros, the Protein Simple Jess, and the Citation 1. You can see that we partner with a number of different organizations, including the Cancer Center, to run these core facilities. The Energy Balance Assessment Core has equipment and expertise for human metabolic phenotyping, small animal metabolic phenotyping, and physical activity, exercise, and meal patterning. This is run by Wendy Court, Ed Melanson, and myself. And we are partnering with the CCTSI and the Department of Medicine and Pediatrics, and of course, the animal facilities to provide these services for people both with clinical research and with small animals, small animal models of disease. Our clinical intervention and translational core is run by Mark Cornier and Janine Higgins, and this is really the core that is run out of the Health and Wellness Center under the we provide services related to the clinical and translational design of research, weight management services, diet composition analysis and nutrition services, physical activity interventions, and a number of various clinical research support activities. And this is with a strong, this core operates with a strong partnership between with the Anschutz Health and Wellness Center and the Colorado CCTSI. So the ongoing initiatives of the NORC, and this is really in partnership with Dan Bessison, who leads the Anschutz Health and Wellness Center. We're investing in a program looking at the science of behavior change. This is particularly relevant or related to adherence to diet and exercise programs and interventions or prescriptions, which is an extremely difficult thing for people to do when they're trying to lose weight or improve their nutrition for health. We also are supporting programmatic development in personalized medicine or tailored treatments for obesity. And this is based upon the Adopt Core Measures project, which is funded or supported in part by the National Cancer Institute. And then what I'm going to touch on in just the few minutes that we have, the disease-specific strategies for better health and wellness. And this is looking for better strategies for particular diseases like cancer. How do we improve these? And so this is where we have an ongoing partnership with the Cancer Center with the Be Fit, Be Well program. Ryan Marker is going to talk a little bit about that, but before I give the floor to him, another part of this is more at the basic science level. I'm going to tell you about the xenograph model that we've developed for studies of obesity and cancer. We call this the DIOX model, the Diet-Induced Obesity Xenograph model. It was originally developed by Liz Welberg, Carol Sartorius, Peter Cavos, and myself. And this was started because most of the transplant models that we use to study cancer are not obesity prone. And so we had to figure out a way to make these animals obese so we could study the relationship between obesity and cancer. And this is developed in the RAG1 null animal on the C57 background. But we found when we study these animals at thermoneutrality, we can make these animals fat, and we can recapitulate obesity-associated tumor promotion with both transformed cell lines and breast cancer PDX cell lines. The model not only makes the animals fat, but we can manipulate the fat and the sucrose in the diet to create lean animals that are unhealthy. So if we give them a high amount of sucrose, we can make them hyperinsulinemic, hyperglycemic, but still lean. And so here, this allows us to look at different aspects of the host's extrinsic factors and how it affects cancer or tumor promotion. You partner that model with the breast tumor bank, like we have with Carol Sartorius and Peter Cavos, and now we have an opportunity to look at a whole host of molecular mechanisms in the tumor, of intrinsic factors in the tumor, and a whole host of factors in the host to dissect out this relationship between obesity and breast cancer. This is just one example of that coming to fruition, where Liz Welberg looked at the role of FGF1 and activated FGF receptor 1 in obesity-associated tumor promotion in the model. It wasn't just the expression of FGF1, but its activation in the tumors that associated with tumor growth and progression. We've also carried these models out and have been studying metastatic disease. Heather Breckbuhl has taken up the reins and is looking at the microenvironment and identified a pro-metastatic extracellular matrix signature that associates with distant organ metastases in this model. My point with actually talking about that model is that we have used it in the breast cancer realm. It is a perfectly good transplant model that we can start studying ovarian cancers or other types of cancers that are associated with obesity, or manipulate nutrition, cancers that are subject to manipulation by nutrition. We have this model now that we can partner with you to help you with these studies, looking at the links between nutrition, obesity, metabolism, and cancer. And so don't hesitate to reach out. The cores are there to help. They know about this pilot program. And I'll turn the time now over to Ryan Marker, who's going to talk to you about the Be Fit, Be Well program, which is a partnership with the Cancer Center at the clinical level that's operating out of the Health and Wellness Center here. So I will stop sharing, and Ryan, are you there?
I'm here.
Okay.
All right. So, like Paul said, I'm going to give a brief overview of the Be Fit, Be Well program that's at the Anschutz Health and Wellness Center. So this is a 3-month exercise program for cancer survivors at the Cancer Center. The actual exercise prescription of this program is highly individualized to each participant in the program. We broadly target improved aerobic fitness as well as muscular strength, but the actual exercise content is tailored based on individual preferences, and as well as any current symptoms that the participant might be experiencing, especially if that participant is undergoing treatment. During the 3 months of the program, participants receive 2 50-minute exercise sessions per week. In the first month of the program, each of those exercise sessions is actually a one-on-one exercise session. And then in the second and third month, those sessions become what we call semi-private sessions, or no more than 4 participants in that session. We perform pre and post assessments, and these include objective measures of physical fitness and physical function, as well as self-reported outcomes that assess fatigue, depression, quality of life, and self-reported physical activity. And participants receive all of this for just a cost of $59 a month, which basically covers their membership to the fitness center. All of our assessments and exercise sessions take place at the Anschutz Health and Wellness Center. And since our participants are members of the center, they get all the benefits, all the other benefits that members of the Wellness Center receive. We've regularly seen significant improvements at the population level in our measures of physical fitness and physical function. So these are things such as the 6-minute walk test and grip strength. And we also see significant improvements in our self-reported outcomes, such as in fatigue and depression. Oversight is provided to the program by Drs. Tom Purcell and John Peters, who co-founded the program back in 2013. I've worked with the program as a postdoc and researcher for several years, but the most important people in our program are cancer exercise specialists. These are all exercise physiologists who work with our participants, but also fulfill various roles within the program. Nicole is our overall manager. Becca is our coordinator and usually the first point of contact for most of our participants. Jared works as a PRA and manages our research database, which I'll talk about in a second, and Ian is our lead trainer. We regularly bring on 2 to 4 undergraduate exercise physiology interns per semester to increase program capacity, as well as to provide that educational opportunity. To join the program, broadly, participants just need to meet 2 criteria. They need to be current patients in the UC Health system, as well as undergoing current treatment or no more than 6 months out of cancer-related treatment. And what we're trying to do is focus our resources on participants who need them the most. Since beginning in 2013, over 600 participants have enrolled in the program, and on average, we enroll 125 to 150 new participants each year. So what research has been done with the Be Fit, Be Well program, and what research can be done with the Be Fit, Be Well program? Like Paul said, with the other resources at the NORC, we're very excited about potential collaborations. We can and have operated as a cancer exercise research laboratory. In the past, we've carried out 2 small prospective investigations, one in pancreatic cancer survivors, and one investigating an intervention to improve physical activity maintenance following the program. And we will serve as a site for the larger tactics study that's run out of CSU once that begins. And finally, we do have an IRB approved research database, which collects all program data for the purpose of future research. This was initiated about 4 years ago, and we have 464 participants currently enrolled in the database. And basically almost all participants that join the BFIP-UL program now provide informed consent to join this database. Our consent has been updated to include access to EMR for various cancer-related variables. And we are currently working on the protocols and procedures to begin collecting blood samples and body composition at both of our assessments.
So Ryan, I'm guessing you're almost done. Oh, never mind. You are.
I am done. Yep, that's BFIT in a nutshell.
If you need to do your acknowledgement, I don't want to cut you off on that.
Oh, that's what I had.
Okay, fantastic. All right, do we have any questions for either speaker? Actually, I'll start off with one for Paul. And I'm sure everyone wants to know, what is it about thermal neutrality that leads to obesity?
Well, we know in the animal models that when we house them downstairs in the facility, they're outside of their thermo comfort zone, and so they're colder. It allows them to leverage their thermoregulatory systems, use their energy expenditure to burn away calories. So it's an opportunity for them to burn away the calories rather than to store them. So studying them under thermoneutral conditions or under their thermo comfort zone, these animals we feel like they're more like humans, and they don't, when we expose them to obesogenic environments, they have a tendency to put on weight just like humans do.
And when you were showing data with the lean controls, was that regular lean, or was that lean? I mean, in other words, was that lean in the normal mouse room temperature, or was that lean that was unhealthy lean?
Those were lean, healthy animals on a low-fat diet under the same thermoneutral conditions. But yeah.
Oh, okay, great. Do we have any other questions from the audience?
I have a quick question. This is Sid Mitra from Pediatrics. I was wondering, have you tried to humanize these mice or develop humanized mouse models within your system, specifically to look at sort of human cytokine responses and stuff?
Yeah, so one of the criticisms of that model is that it doesn't have a fully intact immune system, and the relationship between obesity and cancer could very well have a lot to do with that. So we have not tried to do that yet, but it is in one of our grants that is going out, I believe, next week with Julie Wang, to pursue some of those studies. We have not done it yet.
Other questions? Well, then I'll take the liberty of asking Ryan one. So Ryan, I'm familiar with some studies in mice where they've shown that if you exercise mice after giving them chemo, that there's significant, and in fact, it was actually quite substantial improvement in cardiac function. So I think they were using daunorubicin, which is known to be cardiotoxic, and it leads to long-term impairment of heart function. And they could, I think, almost completely reverse that by having the mice exercise for a period afterwards. Has anything similar been done in humans?
I don't think to that level. We are working with a cardio-oncologist on several projects to look at physical fitness outcomes in patients who have specific cardiac impairments in our program, to try and basically encourage more participants to join to improve cardiac outcomes. But I don't think they've looked at it to that level in humans.
Great.
I have a quick question.
Yeah.
Oh, okay. This is for Paul. Hi, Paul. Have you ever tried in those lean to fat mice, have you ever tried adoptively transferring immune cells into those mice to see how they function?
No, we have not. but it's a good idea, and should be on the list of things to do. So we have not gone that direction yet.
Let's talk about that.
Yeah. And I'll just mention that, I threw little blippets of data up there, mainly for the purpose of letting you know that this model is available. And if, after the retreat, if you have interest, to reach out to me or Heather Breckbuell or Peter Cabos and Carol Sartorius, and we can help, foster some of those interdisciplinary collaborations.
That's
whether
great.
It's related to the pilot project or not. So.
Exactly. And definitely, I think you and Jill and some of the people with the HIMSR should definitely connect in terms of, there was another question also on sort of whether these have been humanized, and that would be sort of a peanut butter and chocolate moment.
Yeah.
So that's great. Thanks so much for the questions. We're going to move on to the next talk. It'll be Dr. Sarah Purcell. And her talk is Assessing the Effects of Exercise on Appetite and Energy Intake in Early Stage Breast Cancer Survivors, Rationale and Design of 2 Pilot Studies. So take it away, Sarah.
Okay, I just need to share my screen. Let's see.
And why Sarah's doing that, Jason reminded me to tell you, if you're on the phone, you have to do *6 to unmute.
Okay, so can everyone see this?
Yes.
Yes.
Okay. So I am a postdoctoral fellow here at Anschutz, and I'm excited to talk about our work on 2 pilot studies we just started in collaboration with my mentors, Dr. Mark Cornier and Ed Melanson, as well as some of the breast medical oncologists and the BFIT Be Well team. So as I'm sure a lot of you know, as screening and detection and treatment strategies continue to improve in certain cancer types, there's an increase in cancer survivors. And in fact, about 17 million cancer survivors are alive in the US, and this is expected to increase. Some of the long-term side effects of cancer and the associated treatments have negative health outcomes, and one of those is obesity. So as shown in this figure of over 500,000 US citizens, the increase in obesity is much higher among those with a previous history of cancer in the dark blue compared to those without a history of cancer in the light blue. And this is especially prevalent in breast cancer survivors. So most breast cancer cases are estrogen receptor and progesterone receptor positive, for which the standard of care is 5 to 10 years of estrogen suppression. And while this is really effective in preventing the cancer from coming back, we know that this is also associated with decreased fat-free mass, which is associated with negative outcomes like quality of life, function, and increased fat mass, which is associated with increased risk of developing obesity-related comorbidities and cancer recurrence. And both of these independently and additively contribute to poor health outcomes. And so to help mitigate the negative effects of these body composition changes, guidelines for cancer survivors for exercise recommend both aerobic and resistance exercise. And this is from the American College of Sports Medicine. I'm not expecting you to read all of this, but just know that for a variety of different outcomes in cancer survivors, both aerobic and resistance exercise is recommended. However, when I look at this, I have 2 questions. And one is the feasibility of consistently meeting both aerobic and resistance exercise over time, and the effect on energy intake and, as a consequence, energy balance, because we know that this is important for the development of obesity. And this is especially important when we consider that 9% of cancer survivors meet physical activity guidelines, and among breast cancer survivors, obesity and previous combination therapy are associated with decreased adherence to physical activity guidelines. However, barriers and enablers to exercise initiation, adherence, and efficacy has been poorly characterized in breast cancer survivors to date. And we don't have a lot of data on dietary intake and essentially no data on how this relates to exercise. But we do know that among breast cancer survivors, we do observe an increase in dietary quality or improvement in dietary quality after treatment. So for example, increased self-reported fruit and vegetable intake, decreased sugar sweetened beverage intake, and about 30% of patients self-report just a general increase in appetite during active treatment. However, we don't know how exercise may influence this. And so I really want to know, with these two studies, what are the effects of exercise on appetite, dietary intake, and energy expenditure in breast cancer survivors with overweight or obesity? And so we're really trying to get at how exercise influences specific components of energy balance. And for the sake of time, I'll just focus on appetite and dietary intake today. But this research question has led to the development of two pilot studies. So we have an acute exercise study called the EARNEST study and a chronic exercise study called the CARE study. Both will enroll 20 women each who have completed active treatment for hormone-positive stage 1 to 3 breast cancer. And these women will have mild obesity or are overweight. And so the EARNEST study, which is the acute study, is investigating the acute effects of resistance exercise on some key markers of appetite regulation. And so the study design is quite simple. We collect measures of body composition via DEXA, resting energy expenditure, and eating behaviors, so things like food cravings at baseline. And then participants are randomized to either a resistance exercise or sedentary control condition first, and they undergo both conditions. And at each study day visit, we assess things like ghrelin and peptide YY, which are important markers of biomarkers of appetite. We also assess ad libitum energy intake, which I'll explain in just a moment, as well as subjective appetite and satiety. And so what each study day looks like, we collect a blood draw at baseline when they arrive, and then ask them about their hunger, as well as desire to eat. And then we feed them a control meal. 30 minutes later, we take another blood draw and collect appetite ratings. They either exercise or rest, and then we collect more blood draws and appetite ratings over the next 2 hours, at which point they're given an ad libitum lunch, which is kind of like a buffet, and then we measure back anything that they don't eat. So we get a good measurement of acute energy intake in response to exercise versus rest. And we think that control to the sedentary condition, we're going to see lower ghrelin and hunger, increased peptide YY and satiety, and an overall decreased energy intake. And so the study is really important for Sorry? Oh, no. Okay. So this
sorry, if you're not Sarah, please make sure to mute yourself. I see Gen 6S Plus, which isn't muted. But yeah, I think there's occasionally we get background noise. Thanks.
So this study is really important for telling us more of the acute response to exercise. If someone works out, what can we expect in the short term? But it doesn't tell us a lot about long term. And so we have worked with BeFit Be Well to develop a program designed to meet the current exercise guidelines of combined aerobic and resistance exercise. And we're assessing some key feasibility outcomes, as well as the impact on the physiological and behavioral components of energy balance, and really focus on the energy intake and appetite part. And so this is a study's design at baseline and follow-up. We collect body composition, resting energy expenditure, and eating behavior. And the appetite and physical activity visits are very similar to the acute study, but there's no exercise condition during the visit. So they just come and they rest for a few hours while we collect measures of appetite and satiety. We're also collecting 3-day diet records and activity monitors after each visit to collect things like physical activity behavior and sedentary behavior. So much like any study, we're also, in terms of feasibility, we're collecting recruitment, enrollment, and adherence, but we're also collecting some information on exercise enjoyment and self-efficacy, as well as safety and tolerability. And we have developed a few study-specific questionnaires to accept things, or to measure things like acceptability during the exercise intervention as well. And much like the acute study, we think that compared to baseline, we're going to see decreases in ghrelin hunger and increases in peptide YY, which will also be associated with decreased energy intake. And we have a lot of other outcomes related to metabolism and physical activity, but for the sake of time, I will leave you with this, and I hope that I've giving you a sense of how important weight management is in survivorship, and how important exercise is in understanding energy balance and how this may impact energy intake and in turn affect energy balance. And hopefully this information can be used to design more interventions for cancer survivors. And so in the spirit of this mini symposium, I just kind of brainstormed some things I would love feedback or help with, or any kind of comments. So the acute study started in late February, and the chronic study was approved by COMAR right in the middle of the COVID shutdowns. But any tips on recruitment would be excellent. The Cancer Center oncologists, as well as BeFit Be Well, have been absolutely amazing in helping me get these studies up and started. But anyone who's been successful in this, please reach out. My background's actually very heavily in nutrition. We've applied to funding to collect some novel analysis of dietary intake, but if anyone has experience on metabolomics or anything related to nutrition, I'd be really interested in hearing that. Biological markers of cancer recurrence would also be important. I don't know if we could expect to see anything over such a short amount of time, but I think that would be interesting to look at too. And then I'm always interested in hearing about translation of practice or anything else. So with that, thank you to my mentors, the cancer research team, and all of you for listening. And I'll leave that up for questions and comments.
Great. Thanks so much, Sarah. We're open for questions.
Hey, Sarah, this is John. Real quick question. It may be a little early in the progress of COVID, but I wonder if there's any information yet about whether people who might lose weight and then get COVID, are they in better shape than somebody who's suffering from obesity and gets COVID? In other words, does weight loss help in the mysterious relationship between obesity and bad COVID outcomes?
I have no idea. I'm not sure. I mean, I think because, it's such short term, I know that weight loss and weight loss maintenance is really, a long-term practice, but I don't know of any studies that have looked at like weight loss maintainers and those who contract COVID. I don't know if anyone else has anything else to add to that, but that's a really good question.
Thanks.
Yeah, of course.
Hey, Sarah. This is Lily. I have a really good talk on and really cool experiment that you're setting up here. My question might be left field, but so your readouts and outcomes are very similar to bariatric surgery, so decrease in appetite, weight loss, and changes in hormones. Have there, just curious, like, has there been any retrospective or any studies on like looking at the outcomes of people who have received these surgeries and then kind of like their risk for cancer afterwards?
I'm not sure because my PhD and current research is more on active cancer and then survivorship. So I'm not really sure like risk, but I imagine when you lose weight, we have all of these favorable metabolic shifts. And so I would imagine, I would hypothesize that their risk would be lower, but I haven't looked into that. That might be an interesting population to look at and to assess, cancer risk and some markers of cancer recurrence or cancer risk. That's a great question.
Yeah, I was thinking like perhaps there's maybe like a common mechanism between the two, and if you can like combine that. I know it's like two different ways to get there, but it would be interesting to see if there's any similarities.
Yeah, and so you're thinking of like specifically just risk, or are you thinking of like people who have cancer and then undergo bariatric surgery?
I guess like for example the hormonal outcomes like decrease in ghrelin, perhaps like that's similar, and maybe instead of targeting, I don't know, like doing bariatric surgery or exercise, you can just go to like changing the hormones level. But I don't really know much about it. It's just a thought.
Yeah, I know that there has been research, even in my supervisor, Mark Cornier's lab, and I can't remember the exact outcomes, but I know that there have been studies that have assessed appetite after bariatric surgery. Now, how that would relate to cancer, it's a really interesting question, because I think that these are both growing populations of people too, cancer survivors and those undergoing bariatric surgeries. So definitely think something to think about.
Thank you.
Sarah, sorry, did I interrupt someone? Oh, okay.
I think
Sarah,
okay.
this is Sid Mitra from Pediatrics again. Again, a question comes from the left field, but this is more for survivors of childhood cancers, and they actually tend to be at very high risk for obesity and cardiovascular disease as they survive, as they grow older. And I'm wondering whether have you thought about developing a program around the pediatric space as well, and I'm just wondering whether that plays a role in their having higher susceptibility as they grow older for recurrence to occur.
I'm really glad you asked this, because I gave a presentation a few months ago and someone asked a similar question, and I can send you an email afterwards, or you can email me. I would love to look at this in childhood cancer survivors, because I think that, as you said, like they are really at risk for obesity to a much greater extent than the average population. And we don't know if that is due to changes in energy intake or changes in physical activity. And when we give these exercise programs, how does that affect all of these downstream outcomes? To my knowledge, very little has assessed this, and I know that our nutrition outcomes are often in 3-day food records, which are not the best at assessing actual dietary intake. So I think that I would love to do a study. It's just a matter of funding and time. But I'll send you an email afterwards, and maybe send you a few articles as well of what I've learned, and then maybe what you've learned as well in childhood cancer survivors.
Thank you. There are actually a number of funding sources within the pediatric space which are heavily focused on quality of life.
Really?
And they might be actually very interested in working with you on this.
Great.
In most of our pediatric conferences, very little is done on post-survivorship.
Yeah, yeah, and I know that they seem to be affected for the rest of their life.
Oh, yes.
Yeah, yeah.
By the way, that's a great connection with Sarah and Sid. So that's something I forgot to mention is one of the things that Cancer Center likes to do is we like to document outcomes from forums and grants and stuff. So if there's any matchmaking or anything that, you know, in other words, if there are any positive outcomes from this mini symposium, we want to know about it. So like Sarah and Sid, if you guys end up, you know, if this stimulates some new project or something comes out of it where, you know, like in the pediatric cancer space, please report back to us.
Will do. Will do.
Yeah. And Sarah, you had mentioned needing metabolomics and, you know, basic kind of resources. Tomorrow, you'll be hearing from Natalie Cercova and Angelo D'Alessandro, and both of them are going to present in, you know, either NMR or mass spec type approaches to metabolomics. So
That'd be great.
tune in.
Absolutely.
We got time for one more question if there's another question out there.
Hi, excuse me.
Hi.
I work on a question in regards to antioxidants. I would like to know if you have any reference regarding obesity and antioxidants focusing on diet.
So are you asking about like just general antioxidants in obesity or antioxidants in cancer survivors or?
Oh, the relation cancer, obesity, and antioxidants.
I'm not entirely sure. I'm not the person to ask. I know that, you know, of course, like more antioxidants are, you know, from the diet, you know, we know that those are associated with several improvements in different health outcomes. But, you know, I think it goes back to the ways that nutrition science has traditionally assessed antioxidant intake is through food frequency questionnaires and 24-hour recalls, which really are not very accurate for that type. I know that there is more research coming out on like metabolomics and that sort of thing to actually measure that. I can't provide a good answer, but I think, you know, this is not the first time I've been asked something about antioxidants in some capacity. It's not really my background, but I think that it's certainly a research niche there that would be really interesting to look at, you know, like more on a population-based study.
Okay, thank you.
I didn't really answer your question. I'm sorry.
Okay, it's fine. Thank you.
Thanks so much, Sarah. That was great. And I loved all the stimulating discussion afterwards. So we need to move on to the next speaker. And the next speaker is, let me see, I moved forward too much, is Dr. Min Han. He's from University of Colorado at Boulder. And his title of his talk is Regulation of Nucleotide Metabolism and Germline Proliferation in Response to Nucleotide Imbalance and Genotoxic Stress by Endo-U Nuclease. Take it away, Min.
Okay, thank you. So I should first mention this is a very basic science right now, and it's still mostly on the metabolism side, but we hope to get somebody interested in to study this in cancer cell. Okay, so in recent years, my lab is interested in a cellular system that responds to nutrient deprivation. Right? So you have certain specific nutrient levels low, you will have defects in cellular process that actually requires the service of a specific nutrient, right? But the animals are often smart enough to build up systems that actually would sense the low levels of the nutrient and then make changes in cellular developmental and behavioral programs to preserve reproductive fitness, if you will, right? So now only these programs on the downstream of the signaling system, not those defect in basic process, would actually can actually be reversed by you perturb the signaling systems. Okay, and then also our focus in our lab is also to study underexplored fatty acids and the nucleotide variants and certain nutrients, and this is about the reproductive system. This is actually the half of the gonad of C. elegans, that's nematode C. elegans, and that's focused on this mitotic zone here, and that germline stem cell will proliferate and will divide and to produce enough cell numbers, and then move on to meiosis before they generate the gametes. And this is perhaps the only system in C. elegans that is used to study cancer biology problems. So Tom Evans in the medical school, at the medical school actually is an expert in this field, right? So, so here's the summary of what my talk is. Basically, nucleotide imbalance or related genotoxicity will have a profound impact on germline stem cell proliferation. And then in the intestine, it goes through this endonucleus, and then it's gonad, and first goes to the NORC receptors that regulates the process. I should also mention that cellular signal system sensing nucleotide changes to dictate the development decision. So many years ago, like more than 10 years ago, we had this hypothesis that perhaps there's a signal system that actually can sense the change in the nucleotide pool in the animal and dictate the germline proliferation, I mean the initiation of germline proliferation. And to do that, you must be able to alter the nucleotide pool in the animal, which turned out to be very difficult to do, right? Because nutrient is coming from food, which is a bacteria involved for nematode, and then also coming from the endogenous metabolic systems. So next, yeah. So you must be able to, no, don't go back one. You must be able to alter nucleotide level and be able to compromise the salvage pathway, right? So next, then we actually eventually figure out this way to do it, and next to actually do bacterial genetics to figure out there's a mutation in the gene that side up, be able to alter the composition there, and also dismantle the salvage pathway by knock out two cytidine deaminases. Next. And by combining those, then you can see a dramatic change in the ratio between cysteine to uridine, and from wild type here to, and the double knockout and uridine level is very low. And next, then we see, next, then germline stem cell proliferation is essentially shut down. From more than 1,000 cells now become less than 50. Animal become 100% sterile. Next. And now, make long story short, Judith Kimball lab and others many years ago have known that NORC receptor controls the initiation of mitotic proliferation. Now we show next that is when the nucleotide uridine level is low and GLP-1 level is shut down and mitotic proliferation is shut down. And showing bottom panel that is all these very few germline stem cells. Next. Now, if you be able to perturb the signal system by creating hypoactive mutation, and using a hypoactive mutation, then you recover mitotic proliferation. As shown, germline production is recovered despite the fact that you still have a low uridine level. So that indicating this is the regulatory system. Then you take them further. Next, that other labs have shown that actually NORC receptor controls the mitotic proliferation actually by shutting down meiosis mechanism and inhibiting two tumor suppressors, the GO1 and GO2. And if that's the scenario next, and then when you lower uridine level, NORC receptor goes down, actually these two gene activity goes up, meiosis going down next. Meiosis goes up and then mitosis is consequently going down, right? So I'll go back one, just sorry. And meiosis goes up, mitosis goes down. And next, and then you should be able to perturb this in total different way now. This time, you actually knock out the NORC receptor instead of hyperactive one. You also knock out both tumor suppressor genes. Then next, then what you see is reversed. Meiosis goes down, mitosis goes up. Then you see this, in this strain contain 5 knockout mutations and germline proliferate like crazy. Okay, next. So that led to the working hypothesis that 4 years ago, there is a nucleotide level in worms will profoundly impact germline proliferation, and mediated by notch receptor with the help of metaconnets. Next question is really about this question marker, and next, Chong Qi in the lab did this difficult genetic suppressor screen, saying, you start out with this double knockout, eating low-yielding food, animals are completely sterile. Now, can you use the mutagen EMS that created suppressor mutations, now convert this animal essentially to fertile? Next, and make a long story short, among the 3 genes we found, one of them encodes indoribonuclease. Next. And here's the data. So in the wild type situation, when this gene is wild type, 100% is sterile, so 0% fertile. Now you knock out this gene, it essentially becomes 100% fertile, right? So you'll be able to rescue the situation by putting the wild type of transgene of the gene behind its own promoter or behind a gut-specific promoter. And you totally reverse that, become 100% sterile again. So that indicating this gene only needs to be expressed in the intestine to do that job. Next. Right? So we quickly found out that this gene actually is induced by the various stress condition. And first is by low uridine level, such as this double knockout by several fold. But next, and we also found that the nucleotide imbalance can be created by adding single nucleotide at a high level, such as 100 millimole uridine, thymidine, or cysteine. You will induce the expression of this gene on the brown bar, right? Next, you can also induce this by using drugs and genotoxic drugs, and that also disrupt the nucleotide metabolism. And you can see that expression also dramatically induced by hydroxyurea, and both is larva and adults. Next. So when this gene is induced under this stress condition, you would imagine this gene probably does something important to deal with such a stress. And that turns out to be the case. So on when the animal is a wild type, you add those single nucleotide to create the nucleotide imbalance, you don't see much problem in the animal growth, right? If you count in the percentage of the animal reach adulthood, everybody is ready 100%. But when you knock out this gene and become super vulnerable, right? So animals are severely retarded, and most animals stay in the early larva stages, and only in this mutant, right? Next. And similar situation with hydroxyurea, and that the damage to animal growth is dramatically enhanced when you knock out this gene, as you see in the orange color here, and all 3 different drugs situation here. Next. And we can also look at the chromosomal segregation defect, and as I can visually, this is called the DNA breach, if you will. That actually, and at the low level in the wild type situation, when you add a single nucleotide such as cysteine, you don't see much changes, and unless you knock out this gene in those 2. When you knock out this gene, then you see dramatically increased damage. Next. And then you have this working hypothesis that is an nucleotide imbalance and condition, a stress condition that induces gene expression. The seems like only purpose is to shut down germline proliferation. And doing so, at least in part, by not prevent animal to come up a salvage pathway, come up with compensatory mechanism. Say, don't try to salvage the situation. Move on to find a better food. That seems to be the protective action here. Next. And then really we're chasing the mechanism question here. We made a critical advance in the mechanism study and showing the role of this nucleotide actually mostly mediated by this enzyme called CTP synthesis that converted UTP to CTP. And we found next that this enzyme gets phosphorylated at the site of serine 532. And next, then this enzyme, the role is actually inhibit this phosphorylation, right? And that seems to does all the trick. And this enzyme phosphorylation is very important. I will show you just 2 pieces of data as an example next. That the bottom band is the non-phosphoryl band, but only when you knock out this gene or using RNA interference, you see the second band, the top band to show up. Right? So what's interesting is that if you use the genetic mutation to totally eliminated germ cells, and you found the bottom band disappeared, but not the top band. And more studies showing that really the phosphorylation, all these things happens just in the intestine, not in the gonad. Next. And I just showed you, knock out this gene, make animal super sensitive to hydroxyurea treatment, and that effect is largely suppressed if you make a point mutation here to changing the serine 532 into alanine, not allow this protein to be phosphorylated. So basically, you increase the non-phosphorylate the protein activity, and then you'll be able to suppress the phenotype. Next. So here summarizes a lot of other data showing that this nucleus actually repress the phosphorylation of this CDP by two independent pathways. One is to inhibit PKA, another kinase, and the other is by inhibit histone deacetylase gene and HDA1, and that leads to the leads to the impact on germline proliferation. Next. So I'm going to first thank the two postdoctors who did this work. When Fanjia is still in my lab, and Chong Qi now is actually at the medical school. The important thing is we hope to do this in mammalian cell next. We actually have evidence that this gene, the ortholog of this gene called INDO-U, actually also induced by hydroxyurea in HeLa cells. And when you knock out this gene, also makes the cells more sensitive to drugs like a 5-FU, right? Of course, this is a very, very preliminary, and the hope is somebody and on the other campus would be interested to study this in cancer cells next. And I'm going to just list all these 5 questions regarding mammalian cell study, and hope somebody will be interested. And if I still have time, I can answer some questions. Thank you.
Great, thanks so much, Min. There's time for some questions. Any questions from the audience? So I'll ask one, Min. What do you, so I mean, obviously you could imagine an inhibitor of INDO-U might be useful to sensitize cells to chemo, but you might also expect that it might increase toxicities.
Absolutely. So that's a lot of unknown. You need to test that. So it's all listed as question number 4 on this slide, which is that's it. We actually have a chemical inhibitor, and somebody want to test it, you can test that, right? So all these 5 things is related using mammalian, perhaps cancer cells. Hydroxyurea is heavily used in certain cancer cells studies, right?
Yeah, for sure.
Yeah. Hello, James. I have a question for Dr. Min.
Yeah, go ahead.
Yeah, great talk. I wanted to ask you, do you see changes with the NDU2 mutations? Do you see changes in the electron shuttles like NADH, NADPH?
We have not. The only other thing I know besides our study is a previous systematic studies when under viral infection, that this gene, not viral, it's the pathogenic bacterial infection, this gene induced 5-fold in the microarray data some years ago.
Thank you.
Great. Other questions?
I have a question, James.
Oh, go, please.
So thank you for that talk. I will reach out because I do have some specific questions, but the one just question that I had is, have you interrogated depth map with respect to the expression of INDO-U and 5-FU's sensitivity in a panel of cancer cell lines?
Yeah, no, we have not done any of these things yet. So really, that's just most of the studies done in C. elegans and just a little bit of mammalian cells, and that the purpose of today's talk. I just really hoping somebody would be interested to do all these things, and that's the one you mentioned to check it out.
So that was Ben Bittler, so you can know who to contact.
Okay. I have a question, James.
Sure, go ahead, Mary.
Okay, so aren't pool imbalances actually mutagenic sometimes? And is that going to be
what's the first word?
Nucleotide pool imbalances, aren't they actually mutagenic sometimes?
We don't know. so I'm telling you, we have not done enough. We use them in animals, you know, basically, yeah, we've not done enough to know because that needs to be done in mammalian cells again.
But yeah, Mary, definitely in other contexts. I know in yeast that's been shown. I don't know how much it's been shown in mammalian cells, but definitely in yeast, it's mutagenic.
Now, mammalian cells, you have data. There's a Cell paper some years ago has showed that nucleotide imbalances cause the chromosome instabilities,
Right.
and that's, you know, related to cancers.
Great. We have time for one more question if someone has one. Going once, going twice. All right. Thanks so much, Min.
Oh,
sorry about
sorry for the mess up of this slides.
You were just trying to be fancy. All right. So we'll move on to our next speaker, Dr. Isabel Schaepfer, and she's here on our campus. And then she's going to tag team it with Dr. Benjamin Bittler, who's also on our campus. And their talk is Fatty Acid Oxidation is a Metabolic Vulnerability in Advanced Epithelial Cancers. So take it away, Isabel.
All right. Thank you to the organizers. Good morning, everybody. And I'm going to tell you, Ben and I are going to tell you about our project together. Next slide, please. So this project and what I've been working on since about 2012, it hinges on the way cancers, benign and cancer cells, use lipids. And to do that, they need to go into the mitochondria. And they use the carnitine shuttle to get in because it's a double membrane. So CPT1A, that is there in labeled in blue, it's an enzyme, it's the rate limiting step for this shuttle. And what it does is that it exchanges the CoA for the carnitine, which allows the fatty acid to travel to the intermembrane space. And then another carnitine palmitoyltransferase, CPT2, in the inner side exchanges now the carnitine for the CoA again, and the fatty acid oxidation can proceed. So CPT1 plays a key role in the flux of this pathway. Next slide. So you would think, what does it do in cancer where there is a strong avidity for glucose? What's the role here? So in this, this is a diagram for a review, but I think it explains well what it might be doing. We don't know exactly. But if you can see in the pink color are the cancer cells that are using glucose and glutamine for proliferation. But in the center in blue, it's the more nutrient-restricted cells that have less access to oxygen and nutrients. And then now they have to switch to fat oxidation to generate acetyl-CoA. So it's very likely that CPT1A plays a role when the cancer is stressed and in conditions like when you hit them with drugs and endocrine therapies. Next slide, please. So the project, I'm going to tell you a little bit about prostate cancer and the work I've been doing there and the role CPT1A, specifically the liver isoform of this transporter. And then my colleague, Dr. Bittler, is going to tell you about our ovary cancer study. Next slide. And going back to that slide, I was telling you about how CPT1 is associated with stress conditions. It happens that when we look in the public databases, on the left, you can see prostate cancer is more associated with aggressive disease. CRPC is castration-resistant prostate cancer. NC is neuroendocrine cancer. So the amplification of this gene is seen there in those stages of the disease. And also there is a significant decrease in survival when you have amplification of the CPT1A gene. The same on parallel on the right, you see ovarian cancer. This is from Oncomine. Stage 4 is associated with more CPT1A expression and also with less overall survival. Next slide, please. So I'm going to tell you about prostate cancer, my side of the story. And for statistics, and according to the American Cancer Society, about 1 in 41 will die of prostate cancer. It's expected for 2020. And the advanced prostate cancer is treated by androgen deprivation, but despite initial responses, most tumors recur. This recurrent disease is termed castration-resistant prostate cancer, and is characterized by active androgen receptor, despite very, very little levels of testosterone. The problem we have in the clinic is that there is no curative therapy for this resistant state of the disease. The only good thing, if there's any, is that this disease has a long natural history and has a dependency on lipids for survival, which is something I've been investigating for a while. Next slide. So what we started doing first is to see if CPT1 was playing a role, if the carnitine shuttle was playing a role in the survival of prostate cancer cells. And this is a biochemical assay to see the palette the cancer cells have for lipids. And in the color bars are metastatic prostate cancer cells, and the black and gray bars are non-tumor forming prostate cells. And you can see from the graph that they have an ability for burning. If I give them radioactive oleate, I can measure radioactive CO2. But when we expose these cells to a CPT1 irreversible inhibitor, atomoxir, with time you see a decrease in the burning capacity of these cells. And when we put them in mice and we do a glucose PET scan, we see something that we expected. If you block the ability to burn lipid, you create, the cells look for another carbon source, which is glucose. So there's a metabolic switch here. You block one carbon source, you increase the uptake of glucose. And indeed, in the lower panel, you can see that an arrow pointing to a tumor that was cold in the basal state, but with 24 hours of atomoxir, you see it brighter, meaning it's taking up more glucose. Next slide, please. One thing that was unexpected is that even though the Warburg effect was activated, with time, if you keep giving the mice systemically atomoxir, you see a decrease in growth, which is shown here by the graph with the red squares. And so this is a pharmacological therapeutic effect. Then we looked at a genetic model by decreasing CPT1A in prostate cancer cell lines, and we saw that there was a decrease in growth. It's not completely eliminated, as you can see in the clonogenic blue dots assay, but there was a significant block in potential for growth, which indicates that by blocking the ability to burn lipid, even though they have access to glucose, it really puts a brake on the cancer growth. Next slide, please. So because I studied prostate cancer, you really cannot think of any therapy without thinking of the one of the pillars of therapeutic approaches, which is the androgen receptor. And one thing I've been studying for a while is this unexpected relationship between lipid burning via the CPT1A enzyme in the mitochondria and the androgen receptor signaling in the nucleus. Excuse me. Next slide. So the next step to try to understand this relationship is to see if there is a gene-drug interaction, if you can increase and decrease CPT1A gene levels and see if there's an association with endocrine therapies. And what we did in this study in mice is that we decrease CPT1A gene. You can see in the upper row, that's control versus knockdown. And you can see in the first panel in A that there's less growth with the knockdown. But if we expose these tumors by a systemic treatment with the anti-androgen and enzalutamide, now there's a more traumatic effect. It's like by targeting CPT1A, we're bringing up more vulnerabilities in the tumors that can make the combination more effective. And interestingly, the opposite was seen when we overexpress the CPT1A gene, when we make the cells And for the sake of time, I don't have time to show you that biochemically, yes, they're burning more lipid and consuming more oxygen, and they're growing better in castration conditions. And when we expose them to the anti-androgen used in the clinic, we see more growth in the cells that overexpress the CPT1A gene. And like I mentioned before, this is a very nice example to show how targeting a metabolic enzyme that is not an oncogene, something that the cancer cells co-opt for their survival, it can open vulnerabilities to other therapies like chemotherapy, which is something my colleague, Dr. Bidler, is going to tell you right now. Thank you very much for your attention.
Next slide. So thank you, Dr. Stuffer, for that wonderful introduction into CPT1A and your work in prostate cancer. And so I just want to take two steps back, and then I'll talk about how we got involved with Dr. Stuffer's work in CPT1A. And ovarian cancer is my major area of interest, and we are really focused on how ovarian cancer disseminates and how it progresses as a whole. But as you can see here, most ovarian cancer actually doesn't arise in the ovary. It actually arises in the fallopian tube and then disseminates to fat-rich organs. In this particular case, we have the omentum, which is a very fat-rich environment. With respect to why we need ovarian cancer therapeutics is largely because outside of chemotherapy and PARP inhibitors, they're very limited with respect to targeted precision medicine approaches to target ovarian cancer beyond failure of chemotherapy and PARP inhibitors. So there's definitely a need to develop new therapies for ovarian cancer. Next slide. So to model advanced ovarian cancer and really to advance disease, what we did is we performed a synthetic lethality CRISPR-Cas9 screen here using the functional genomics facility, as well in parallel transcriptomic analysis. And so this is a relatively complicated slide to digest. And so I just want to go through it a little bit here. And so this is some of the major metabolic pathways that we found to be involved in ovarian cancer dissemination as well as advanced disease. And what you can see here is, if you go to the next, just click next slide. So what we found is the red bars and the red boxes and ellipses indicate genes and metabolites that were enriched as the ovarian cancer is disseminating, like I said, as a model of advanced disease. And what we found is a very highly enriched pathway with respect to carnitine and fatty acid metabolism. And so then we started to do a little bit of research and literature searches on this particular pathway, and that's when we discovered Dr. Stuffer's work here in prostate cancer, and that was where this particular collaboration was born. And so while we're interested in fatty acid oxidation, you can see from this particular figure, there are other metabolic pathways involved in ovarian cancer potentially driving the disease to progress. So next slide. And so all of this work was published earlier this year. So I'm not going to belabor it too much, but this is work done by a very talented clinical fellow, Brandon Sawyer. And what we found was that on the left here, we're using an ovarian cancer cell line. We use SH control and SH-CPT1A, similar to Isabella's work. And what we found is when we put them in suspension, you can see in the gray bar, we reduce the maximal respiration measured on the y-axis there. However, the addition of an exogenous fatty acid or long-chain fatty acid, in this context, it was oleic acid, we can rescue that metabolic stress. What we did then is we put these SH-CPT1A knockdown cells and found that when you put them in suspension or anchorage independence, you actually it can exacerbate that metabolic stress. And the addition of oleic acid beyond that did not rescue it back to what we were seeing in the adherent conditions, suggesting that these cells are more dependent on CPT1A and to metabolize long-chain fatty acids for ultimately to reduce their metabolic stress. And similar to what Isabel had demonstrated, we combined atomoxir, which is the reversible CPT1 inhibitor, and found that we're able to significantly reduce tumor growth over a 21-day treatment schedule. This was combined with cisplatin, and fortunately or unfortunately, these particular PDX models were highly sensitive to cisplatin. But what we did notice was that in the cisplatin etamoxer group, these particular mice didn't lose as much weight, and they actually were healthier at the end of study, which is an interesting finding. Next slide. So these two pieces of work from Isabel's and my lab really are the foundation for this particular pilot award that we intend to apply for. And really what Isabel had demonstrated in prostate cancer is this relationship between AR signaling and CPT1A, as well as, I'll show you in a second, a description of a novel axis between CPT1A and PI3 kinase AKT. In ovarian cancer, we showed, and this is a little bit more in its infancy with respect to Isabel's work, but we found that CPT1A upregulates fatty acid oxidations to survive in a more stressful environment, potentially under chemoresistance as well as the metastatic process. So next slide. So these were the two real major questions that we wanted to approach in our pilot award and ultimately as we develop into a larger federally funded award. And so to examine the crosstalk in both prostate and ovarian cancers, as well as triple negative breast cancers, potentially through other collaborations on campus with Dr. Jenberger. But to look at this axis of CPT1A signaling and the insulin, PI3 kinase, AKT, mTOR signaling axis. And then also one of the things that we get a lot from respect to our clinical collaborators is, trying to identify the potential of contribution with their chemotherapeutic response. And I appreciate some of the earlier talks with respect to starting to address some of these, and I appreciate potential of future collaborations. So next slide.
So Ben, you're cutting into your question time right now, just so you know.
Ever so quickly, so we found that loss of CPT1A regulates, and this is unknown as to how this regulation occurs, a phosphatase that mimics, it mimics P10, and so we found that the loss of CPT1A, you lose phospho-AKT. So this is the contribution of CPT1A and phosphatase PI3 kinase in this prostate cancer that we hope to interrogate. Next slide. Next slide. Next slide. So we have three aims. One is a hypothesis-driven aim to appreciate this PI3 kinase AKT signaling axis with CPT1A, as well as hypothesis generating to do a small molecule screen looking at long-chain fatty acid exposure and chemotherapy response, as well as translational implications. And I really appreciated Paul's presentation at the beginning, because I think both Isabel and I would be interested in using the DIOX model for this aim 3. Next slide. So we've already identified several clinical and basic science collaborators, as well as shared resources that we intend to use. However, this is our call to help. If anybody has any added advice for us or wants to be a part of this project, we are looking for collaborators. Next slide. And so just quickly acknowledging the people that have done this work, Brandon Sawyer, Lindsay Wheeler from my lab, Isabel Schlepfer, and her lab and the contribution, and yeah, you can see the people listed there. So thank you for your attention.
Great, thanks to both Ben and Isabel. We have time for a few questions, particularly if they're fast questions.
So I see Haida's question. The answer to your question is they mostly move away in clumps. So when we see ascites fluid, we see large clumps of cells that are attached to each other, not so much single cells.
So that's what I thought. So I'm wondering, is there a way you can mimic that in your sort of suspension assays? Because your suspension, I would think, would be more single cell, and metabolically they may be different if they're in clumps versus single cells. I don't know.
So that's a great question. So when we put them in, they are in single cell suspension, but then they clump together. And we have beautiful images showing ascites, ovarian cancer cells, and suspension ovarian cancer cells, and they look identical. They form these nice rafts.
Oh, that's great.
Yeah.
This is Ed Melanson, and I have a quick question for Isabel. But first, Isabel and Ben, great presentation, wonderful data. Isabel, thinking from an integrated perspective about exercise, where you're mobilizing free fatty acids, but yet they're being taken up and oxidized in skeletal muscle. Have you thought about how exercise integrates into this picture?
Yes, actually, I quickly, I don't know the complete answer to your question, but I could speculate that exercise will pull away, fatty acids from the circulation and perhaps pull them away from the tumor. So it will be a way to restrict the tumor from lipid burning capability. I would envision dietary and exercise approach in combination with the treatment for tumors that have excess CPT1A. That might be actually a better outcome.
Thank you. That's interesting. Thank you.
Can I ask one more question, James?
Okay, quick on this one. Go
Okay,
for it,
just
Natalie.
very quick. So just continue on ask question. Isabel, you said the dietary restrictions could also help. Which one would those be? I mean, do you have any ideas?
Yeah, well, that's something I discussed with Ben. It's diets that will reduce insulin and glucose. Just briefly, he mentioned the AKT connection with CPT1A. I envision, for example, a low-carb, high-fat diet. I think that will result in reduced glucose and reduced insulin, and it will probably starve the tumors from fatty acids.
What about
to make ketone bodies to sustain the whole body metabolism?
Okay, and do you think something like metformin will help also?
Yeah, I think that is something to consider. I worry that metformin activates AMP kinase and may promote fat oxidation in the tumors, but I haven't tested that. That's a good point, Natalie. I haven't tested that. I just speculate that it
Okay, thank you.
I see one expression. Thank you.
Thank you.
Great, thanks everyone. So we've gone from humans and exercise to worms, now to mice, and now we're going to go to small molecules. So the last speaker is Phil Regan. He's in the School of Pharmacy and Pharmaceutical Sciences, and he's going to be talking about targeting the AMPK RSK axis in acute myeloid leukemia. Take it away, Phil.
All right, so I might need Jason to help me in a minute, but we'll see if we can share the slides. Okay. Can we all see them?
Yep, looks great.
Cool. All right. So I'm an associate professor at the School of Pharmacy. I'm a medicinal chemist. We try not to go down the biological vortex too much. We normally leave that to collaborators. And so what we try and do is develop chemical tools to answer biological problems and hopefully develop a drug candidate from that. What's kind of been interesting, we've kind of had this risk inhibitor project going in the lab for a couple of years now, and we've also had an AMPK project going in the lab with a couple of years. The AMPK project's in collaboration with Craig Jordan, and but it's been kind of interesting that over the last 4 or 5 years, there's accumulating evidence that both AMPK and RISC may be important targets for eliminating cancer stem cells or even sensitizing cancer stem cells to conventional therapy. And it's kind of puzzling to us from a chemist's point of view anyway, because RISC is a downstream effector of the RAS-RAF-MEK pathway. And it does have some kind of crosstalk with AMPK, but mainly through LKB1, which is a major activating kinase of AMPK. But actually RISC inhibits this activation. And also RISC has a whole plethora of targets, but most of them work in opposition to AMPK. So it's kind of interesting that you have, especially in leukemia, RISC is normally, or the RAS-RAF-MEK pathway is normally highly activated in leukemias, but there's also reports now that AMPK expression and activation is also important in these cancers. And so it's kind of an interesting kind of dilemma of which one do you target? Do you target one or the other, or do we target both? And there's been more recent reports, which we'll go on to in the talk, to maybe describe how we could approach that. The main interesting downstream targets that we're interested in terms of risk is things like MCL1, BAD, GSK3. We're not necessarily interested in LKB1. There's a lot of researchers in that area, and we tend to want to stay out of it. Also, Krebs, obviously a major target in leukemias as well. So with AMPK, the previous talk kind of talked about AMPK briefly, but basically it's a kinase, and it regulates a lot of downstream targets in times of metabolic stress. And it's regulated by low ATP levels. So under those low ATP levels, AMPK is activated, and it's like a thermostat, but it's actually trying to regulate energy processes to try and readdress a homeostatic balance. So what was really interesting is studies by the Jordan lab, and there's other studies out there, but the Jordan lab used reactive oxygen species as an indicator of metabolic activity in primary AML specimens. And they found that in this ROS low population, the leukemic stem cells tended to hide out in this ROS low population. They tended to kind of basically prefer that kind of environment. And that environment was also relying on oxidative phosphorylation. These cells were relying on this process for survival. And the other kind of interesting thing that they found was knocking down AMPK resulted in an increase in ROS levels and a loss of leukemic stem cell viability. So it basically started to accumulate evidence that inhibition of AMPK in the cancer stem cells may be a nice target, particularly in the fact that Craig's group also looked at knocking down AMPK in normal hematopoietic stem cells, and it really didn't affect their viability at all. So unfortunately, or fortunately for me, there are actually few AMPK inhibitors. Compound C is frequently used, but it has broad spectrum activity. It actually inhibits other kinases more potently than AMPK. And also this other compound, we call it SBI, but the IC50 values are fairly poor, and they require really high concentrations to inhibit AMPK in cells. One compound that does inhibit AMPK very potently is sunitinib, but of course you probably all know that sunitinib has broad scope kinome activity. But the interesting thing with sunitinib, because it does have the potency and it does have this oxyndole ring system, it gives us a nice synthetic platform to start developing compounds to try and optimize those interactions with AMPK, to try and maybe dial in further potency, but more importantly, selectivity. And so you can kind of see we got the computer cranking, we get these crystal structures out from the protein data bank. We're then using a computer to kind of virtually build our molecules inside. We can then strategically look in the pocket and say, well, there's a hydrophobic pocket. We can build towards that hydrophobic pocket, but more importantly, build towards this DFG motif, which in kinases can introduce both selectivity and potency, and that's kind of what we did. We also changed out this alkyl amino side chain as well, which is basically has important anchoring interactions on the periphery of AMPK. Results are kind of shown on the right of the screen here. We made about 25 compounds initially. Some were more potent than sunitinib. CM25 was more selective. We published recently, if you want to look at all that kinome data. Because sunitinib hits VEGF-R, we also did a KDR assay as well to look at the full selective difference in one of its primary targets. So we took things a little bit further. So the data on the previous table was data from a TR-FRET assay, which is using recombinant protein. And we wanted to look at cell engagement as well. Unfortunately, with sunitinib, and it can be a positive and a negative, these types of compounds have fluorescent activity, which can be great for imaging, but it's very poor when you want to do any kind of FRET-based assays. The TR-FRET assay was okay. We seem to be out of the range there in clear space, and we could do that assay with fairly nice error bars. But when we went to do a nanobret, which is kind of like an easy chemist cell engagement assay, it basically the properties of the compound interfered with the assay. So we had to go to an ELISA. So we looked at the ELISA assay, and if you kind of remember back to the previous slides of the known AMPK inhibitors, they were all inhibiting at about 30 micromolar in cells, and we were getting pretty good inhibition at about 5 micromolar. You know, we could really knock it on its head with 50 micromolar in the ELISA assays. But one interesting thing that we found was sunitinib is a very potent cytotoxic species, but the AMPK inhibitors, we dialed in some selectivity. We completely eradicated their activity against tyrosine receptor kinases, and they seem to have really no impact on cell viability. And we'll come to that in the next slide. Another interesting thing that we found was a compound, CM228, wasn't very potent in the TR-FRET assay, but actually was quite potent in the ELISA assay that I just showed on the previous page. And this kind of lends an approach to inbuild some kind of selectivity. AMPK is basically a trimeric structure of 3 components: an alpha component, which is the catalytic component, the beta, which is kind of a bridging component, and a gamma, which is the regulating component of the protein. So the alpha catalytic component is what we're targeting here, and it has 2 isoforms, an alpha-1 and an alpha-2 isoform. The alpha-1 isoform is present through the body, but this seems to be the common or the predominant form in leukemic stem cells, whereas the alpha-2 isoform is expressed in the heart. And these 2 isoforms have about 90% homology. So targeting one over the other with an isoform selective inhibitor is going to be highly unlikely. So we think a way of getting around that is by developing a hypoxic or a hypoxia-activated prodrug of 228. And so these leukemic stem cells tend to reside in the bone marrow microenvironment where it tends to be hypoxic. And so this is an attractive feature for chemists to exploit in terms of drug delivery and introduce some selectivity into the AMPK inhibition. Now, the targeting of the alpha-2 isoform in the heart is being proposed to be part of the mechanism of sunitinib's cardiotoxicity, but obviously sunitinib is used in the clinic, and so that cardiotoxicity is probably manageable. But we kind of think that this could be a nice approach to maybe target a feature and introduce some selectivity. And we're currently looking at bioactivation in cell systems of this agent. Since this is kind of a metabolic talk, I kind of wanted to introduce this because as chemists, we kind of like to develop a compound where we get, or we can show cellular engagement of target before we move on to all those DMPK experiments. So we kind of want to know that we've developed a compound that can get into cells and can hit the cellular target of interest. And to do that, we kind of had to play around with the cell systems. It's a common feature when you're targeting AMPK to actually grow the cells in low glucose media, which we did. And we've kind of flanking leukemic lines with Miopaccus and HepG2s, which we grew in known conditions to express and activate AMPK. And you can see in the leukemic lines, there's a lot of expression of AMPK in low sugar media, and there's a lot of activation there as well. So we're kind of pushing the activation, which is great to kind of show how important is AMPK inhibition. Interestingly, when you look at the MOLM13s, which is what we used a lot of, and the K562s, but when you look at the MOLM13s, when you grow them in sugar media, there's still a lot of expression and activation of AMPK, but there isn't in MOLM14s. And like kind of looking retrospectively back, we kind of think that maybe what we should have done is challenge the MOLM14s with the AMPK inhibitors, because it could be in the low glucose media, the more reliant on AMPK. And so that's something we're looking at doing currently as well. AMPK and other cancers, of course, targeting cancers with, or challenging cancers with metformin, ACAR is something of interest to kind of push the balance the other way with AMPK and activate it. But there are kind of other studies that are kind of showing that targeting stem cell components with AMPK inhibitors is kind of the way to go. And in glioblastoma, there's been a recent report where they looked at normal human astrocytes and challenged it with shRNA, and basically didn't impact their viability, but it did impact basically glioblastoma stem cell-like cells over time. And as you can see from the days, they're going out to about 5 days and they're only getting down to about 40 to 50% viability. So it kind of dovetails with our experiments where we're not really seeing a huge impact or an acute impact on AMPK inhibition in leukemic cells. Interestingly, very recently, a large consortium of researchers published in Cancer Cell, and they kind of published that venetoclax, a BCL2 inhibitor that's frequently used in leukemia treatment, has these mechanisms of resistance, not attributed to mutation of BCL2, but attributed to increased activation of AMPK and expression of MCL1. Well, MCL1 is actually regulated by RISC, and so we kind of think that maybe through inhibition of AMPK or RISC, we might actually eradicate this mechanism of drug resistance that seems to be being observed in these patient samples. And of course, that's something that's kind of of interest to us right now. And of course, the Jordan Lab are always looking at combinations with venetoclax. So that's something we're kind of planning on doing in the future also. With the RISC inhibitors, I should show this other cartoon, and this is one I stole from the Cook et al. Paper, but just to show that it's kind of, MCL2 is kind of, in the BCL2 family, are kind of tightly associated to ERK and RISC. So it's kind of interesting, again, that we have RISC, a downstream effector, and AMPK kind of as kind of culprits as maybe a mechanism of resistance, but when activated, these two proteins kind of do opposing things. So it'd be interesting to challenge them with small molecule inhibitors with venetoclax. With our RISC project, unfortunately, I can't really discuss much of the RISC project, which is unfortunate. We're just a little bit too late in terms of scheduling this conference because my patent attorneys are finishing the patent on July 6th. So we can't really describe the latter compounds. But what we did find, we started off with this compound BID1870, which is a known RISC inhibitor. We did exactly the same thing that we did with sunitinib. We changed parts of the molecule, excuse me, to try and optimize it against RISC. And we found that we could get RISC inhibition in cells without that concomitant kind of cytotoxicity. We looked at cytotoxicity and cytostatic effects as well, and there's really no effects over 72 hours. It's kind of a really interesting feature that we can take out either RISC or AMPK and really not have that much of an effect on cell viability. Now, we expect that there's other challenges here. It might be that we need to grow the cells up in a hypoxic environment to really kind of recapitulate the environment of the tumor and maybe make the cells more reliant on oxidative phosphorylation, make the cells more reliant on an AMPK pathway. We also think that there might be a pivotal, this is why we kind of call it the AMPK RISC axis, because it's kind of interesting that AMPK suppresses protein synthesis while RISC promotes protein synthesis. So we could be looking at two protein targets that are kind of a pivot point between the stem cells being quiescent to then driving tumor formation. I don't know. I'm a chemist, I'm not a biologist. But we're developing 3 additional approaches to target AMPK and including targeting molecules that bind to the regulatory gamma subunit. And these could be kind of interesting because we could change the molecules very subtly and we could have an activator of AMPK or we could have an inhibitor, and we're kind of excited about that. We're also establishing methods to evaluate the bioactivation of that hypoxia-activated prodrug, and we're looking at those venetoclax combinations as well. With the RISC inhibitors, we're looking at the 2nd and 3rd generation inhibitors right now, and we plan on doing the venetoclax combinations there as well. The summary, I'm not sure how I'm doing on time, but I think at present what we found is pharmacological targeting of AMPK or RISC with a small molecule inhibitor doesn't really appear to impact.
Phil, you are into the question time, so you may want to go through these quickly.
I will do my last point then. But these compounds don't really impact cell viability. And it's kind of interesting that both pathways intersect upon one another, but they have opposing effects. The venetoclax publication is very interesting because the kind of whole plethora of downstream targets for RISC has had us scratching our heads in terms of looking at cell engagement. But now maybe what we should be looking at and tuning into is MCL1 and CREB for looking at downstream targeting of risk. And hopefully, we will be able to identify ultimately an optimal targeting strategy to basically suppress venetoclax resistance. And then lastly, thank the collaborators, my lab, and of course, funding sources.
Great, thanks so much, Phil. We've got time for a few questions. So Phil, the MOM13 and MOM14s, they're derived from the same leukemia, but they're two different clones. And the MOM14s are more monocytic-like, which is thought to be more resistant to venetoclax. So your results are really interesting from that perspective, and that you see such a striking difference in the response of those pathways to glucose.
Yeah, I think this was kind of, we were very eager to really challenge a cell line that expressed AMPK or expressed activated AMPK, and that's why we went with MOLM13s. And like I said, in retrospect, it would have been great to maybe challenge both cell lines because of the differences here in terms of their activation of AMPK and their status with AMPK, and obviously challenging them in the two different environments, plus minus glucose. It might have given us an insight into mainly MOLM13s grown in glucose deficient media might be more susceptible to AMPK inhibition in terms of their viability. So maybe we're missing something there.
Yeah, I mean, really, the oddballs are the K562 and the MOM13s because they don't appropriately shut it down with glucose. So
yeah,
cool. Do you have any other questions from the audience?
Yeah, let me turn my video on. Yeah, I was wondering, your inhibitors are targeting the alpha-1 and alpha-2. Is there any chance or potential of targeting the beta or gamma subunits to modify activity or modify the regulation of the activity?
So it was interesting because the glioblastoma paper, which I presented a little bit of the viability data, they actually did an SH RNA knockdown of the beta unit. So the beta unit kind of connects the alpha and the gamma. And so they knock that out, and so you don't have functional protein. So that's kind of interesting, but the beta unit really is just a bit of strand of protein that kind of links the two alpha and the gamma together. So there's nothing really targetable on that stretch of protein, unfortunately. The gamma, like I said, is kind of really interesting to us because there's 3 AMP ATP sites there. And of course, when ATP levels are low, AMP binds to that regulatory site, changes the protein's conformation, activates AMPK. So we kind of think that's kind of interesting because there's 3 sites there. They have subtle differences in their composition. And so we could target one over the other and we could get activation, or we could target one over the other and we could get inhibition. And it might also introduce a level of selectivity as well, because they don't typically look like the normal kinase ATP site. And that's normally the problem with targeting kinases. Everybody goes to the ATP site, which is kind of borderline on the kinase domain because you're transferring that phosphate. But if we kind of target the gamma domain, we think we might be able to get some clear blue water selectivity there. And we just have a backlog of compounds that are kind of in testing right now. So, yeah.
Thanks.
All right. Well, I know in lieu of other questions, first, let's thank all the speakers. If everyone could clap, they would. Great talks, and really it shows the sort of the breadth of different research that's going on in Colorado related to nutrition and cancer. And hopefully this will stimulate some interdisciplinary interactions. And of course, join us tomorrow, 2 p.m. for the second half of the symposium. And you've all got the agenda, so I won't go over it, but it goes from 2 to 4. So we hope you all join us then, and everyone have a good rest of your day. Thanks so much.